Good day, everyone. I am Christine Joy L. Paliagaw. Together with Janina Molina and Laika Concepcion, we are the reporters for today's session. Last time, we talked about the little genes and the pseudo-allelism that being explained by the former group. Today, we're going to talk about the environmental influence of gene segregation, the external and internal factors that affect the gene segregation. Under external factors, we have the temperature, light, nutrition, material relations, and lastly, the drug and chemicals. And under internal factors, we have age, sex, sex influence, sex limited, and substrate that we're going to explain in the next slides. The expression of genes in an organism can be influenced by the environmental factors. This includes the external world where the organisms develops or located, and also the internal world, which includes factors such as hormones and metabolisms in our body. The external world is a person's subjective sense of self and all of the thoughts that it generates. The external world is everything else. The internal world is everything you see or experience, and the external world is everything causing what you see and what you experience. Now we're going to talk about the external environmental effects. The external environmental effects are those leading to phenotypic changes that seem directly correlated with observable changes outside the organisms. Under external environmental effects, we have temperature. There is a close correlation between the rate of chemical reactions and temperature. Since the basic developmental effects of many genes is to control the rate of a specific reactions, a change in temperature can be expected to have a widespread effect on development. The example in this is the species of primrose plants with varied flower colors. The red color of the flower primrose is at room temperature, while its white color is at temperature over 30 degrees Celsius. And another example for this is the Himalayan rabbits carry the C gene, which is required for the development of pigments in the fur, skin, and eyes, and whose expression is regulated by the temperature. Specifically, the C gene is inactive above 35 degrees Celsius, and it is maximally active from 15 degrees Celsius to 25 degrees Celsius. This temperature regulation of gene expression produces rabbits with a distinct coat coloring. And as you can see in the picture, the white rabbit with black spot is reared at 20 degrees Celsius or less, and pure white rabbit is reared at temperature above 30 degrees Celsius. Next is we have the light. Energy provided by light and light itself are essential for the growth and development of plants and animals. For example, seedlings grown in the dark do not survive long because chlorophyll is not developed, even though there are genes for chlorophyll development. Varieties of sweet corn can be an example for this. In plants with this gene, the parts exposed to the sun are red, but those in shaded areas are green. Light can also influence gene expression, as in the case of butterfly wing development and growth. For example, in year 1917, biologist Thomas Hunt Morgan conducted studies in which he placed Vanessa urtica and Vanessa ayo caterpillars under red, green, or blue light while other caterpillars were kept in the dark. When the caterpillars developed into butterflies, their wings showed dramatic differences. Exposure to red light resulted in intensely colored wings, while exposure to green light resulted in dusky wings. Blue light and darkness led to paler colored wings of the butterfly.
Nutrition. Food provides energy for carrying out necessary processes and materials that must be incorporated into necessary structures. Genetically, different organisms, even of the same species, usually differ in the kind and amount of nutrients they require. Depending upon its genotype, simple dietary changes can lead to different phenotypes. Example, the appearance of yellow fat in rabbits depends on genotype YY and green vegetables eaten. Elimination of green vegetables also eliminates the appearance of yellow fat. Next is material relations. In case of mammals where the direct body relations between parent and progeny extend beyond fertilization, interaction between the fetus and the maternal environment occurs. For example, blood group incompatibilities between the mother and offspring produce special effects on the survival of particular genotypes. One example also is a maternal infection during pregnancy. It is therefore unsurprising that even a slight environmental insult during these extensive developmental processes can lead to long-lasting structural and functional alterations in the offspring. Next is drugs and chemicals. The presence of drugs and chemicals in an organism environment can also influence gene expression in the organism. Cyclofish are a dramatic example of the way in which the environmental chemical can affect development. In 1907, researchers Stockard created cyclopean fish by placing fertilized Fundulus heteroclitus eggs in 100 ml of seawater mixed with approximately 6 grams of magnesium chloride. Normally, Fundulus heteroclitus embryos features 2 eyes. However, in this experiment, half of the eggs placed in magnesium chloride mixture gave rise to one-eyed embryos, in which he called it cyclope fish containing one eye.
So we are done with the external environment that influences the gene expression. Now let us proceed to the internal environment. So internal environment effects are those leading to phenotypic changes that appear correlated primarily to the changes within the organism. Some examples appear correlated primarily to the changes within the organism. First is the age. As an individual grows older, there are certain phenotypic changes that occur which allow further genotypic effects to be expressed. For example, is the age of onset of different genetic traits in human. So as we grow older, may mga bagay na dinedemand ang ating katawan para mag-grow. Second is sex. There are two categories or two types of this. First is the sex-controlled or sex-influenced, and the other one is sex-limited. In sex-controlled or sex-influenced, hereditary traits are more common in one sex than the other, such as hair lip, pattern baldness, and gout, occurring more frequently in men than in women. On the other hand, spina bifida, forked spine with open spinal cord, is found more frequently in women than in men. Sex-influenced traits are those that are expressed differently in the two sexes, such as traits are autosomal, which means that the genes responsible for their expression are not carried on the sex chromosomes. For example, is the male pattern baldness. Napapansin natin na mas frequent or mas nakikita natin yung pagkakalbo sa mga lalaki rather than sa mga babae. Dahil yun dun sa it is influenced by the hormones testosterone and dihydrotestosterone, but only when levels of two hormones are high. So hindi naman sa lahat ng lalaki nangyayari yun. Yun lang ay kung mataas yung level or mataas yung kanilang testosterone and dihydrotestosterone. In general, males have much higher levels of these hormones than females, so the baldness allele has a stronger effect in the males than in females. Posible din naman na magkaroon ng baldness sa babae, yun lamang ay kung mataas ang kanilang stress level. So, ang stress talaga ay nakakapagpabago ng gene hormones. Next is the sex-limited. Sex-limited appear in one sex and not in the other. Genes that influence milk yield in humans and dairy cattle. Sex-limited traits are also autosomal. Unlike sex-influenced traits, whose expression differs according to sex, sex-limited traits are expressed in individuals of only one sex. So, ito ay applicable lamang sa iisang sex. As an example of a sex-limited trait is the lactation or milk production. So, limited lang yung pagpuproduce ng milk sa mga babae. Lastly is the substrate. The kinds of reaction that takes place in an organism depend largely on the substrates present. These substrates are synthesized by the organism, and their presence or absence may be genetically controlled. For instance, in diabetes mellitus, sugar levels normally controlled by the insulin are not properly regulated, resulting in high blood sugar level and excretion of sugar in the urine. So the external and internal environmental influence on gene segregation has a great effect on the gene segregation or in producing a new offspring of a certain product. Well, these are the factors that can be considered in observing the developmental process of an organism or an individual. The phenotype of an organism is the product of interaction of its genes and environment. The genotype provides the messages and direction for phenotypic growth and behavior, and the environmental factors must be supplied so that the organism may develop and continuously exist. This is our report on environmental influence of gene segregation. Next is the probability